Hi, my name is Chloe Goebel, and this is my research project. Um, I did my protein project on Sonic Hedgehog protein, and uh, the ID for that is 3HO5, just what I used to research it. Just an ID number, and SHH is what I'll be addressing it as for the rest of the presentation. So just to get things started, you guys can think about how the two of these things are related. Very weird, but Sonic Hedgehog and embryonic development, they actually contribute to the function. So the SHH function functions in embryonic development. So it plays a key role in the developmental aspect of the skeletal system and the nervous system. And if there is an abnormal signaling or dysregulation of the SHH pathway, it can actually result in several types of cancers, like the ones I have listed on the slide. So gastrointestinal, ovarian, brain, lung, skin, prostate, and breast cancer. And I chose this protein because I think it's really interesting about how important it is to our development, and how like a minor dysregulation of the pathway can lead to such severe diseases in our bodies. And I guess the pretty obvious one was the name. It's funny and unique. So let's get into the structure a little bit. So the SHH protein actually has two different subunits, A and B chains, which are the blue and the green, are made up of that sequence, while the C chain, the purplish pink one, is made up of a smaller subunit with that sequence. And this actually makes the primary structure. The individual subunits link together via peptide bonds and create the primary structure. Then the secondary structure is when, as we know, alpha helices and beta pleated sheets come together via hydrogen bonds, link together, creating the secondary structure of the protein. The tertiary structure is each individual color, so the green, the blue, and the pink. And each of those tertiary structures forms by their alpha helices and beta pleated sheets collapsing in on one another by hydrophobic collapse and through their R group interactions. And the quaternary structure forms as the three tertiary structures, three individual tertiary structures, come together via hydrogen bonds, creating the big happy structure and protein that we see on the screen. So the disulfide bridges are highlighted in the image above. They're kind of hard to see, but they're there. Most of them are in green. But the disulfide bridges are cysteine residues that just help stabilize the tertiary and quaternary structures of the protein, just giving them more stability. And the function related to the structure, as of right now, researchers haven't figured out how the structure of the SHH protein relates to its function. I was reading a 2020 article that stated the mechanism of SHH signaling is complex and remains incompletely understood. So hopefully in the future we'll have an explanation of how the structure relates to its function, because it is very important in our development. And to end, I just have some fun facts. Mammals have 3 HH homologs: desert, Indian, and Sonic. Sonic being this protein, and the most heavily researched HH homolog. And the HH pathway is heavily researched for potential cancer treatments, because like we talked about, a simple dysregulation can cause cancer. So it's just a really good start for researchers. And lastly, the SHH protein was originally identified in the common fruit fly by two developmental biologists and was officially published as a protein in 1980. So thank you very much.